Welcome to this explainer. Designing a perfectly specific guide RNA and producing the most efficient Cas9 variant in the lab is an incredible feat of biological engineering. But here's the stark reality. It means absolutely nothing if you can't get those molecular components inside the nucleus of your target cell. Because ultimately, solving this specific bottleneck is the master key to actually curing genetic diseases in human patients.
Think of the cell's plasma membrane as basically an impenetrable fortress. It is specifically evolutionarily designed to exclude large, charged macromolecules. By default, proteins, DNA, and RNA are all fiercely repelled at the gates. So how on earth do we sneak our perfect Cas9 past the guards?
Okay, let's dive into this. Here's our roadmap for demystifying this massive genetic bottleneck. We're going to cover the delivery problem, the 6 major platforms, do a deep dive into comparisons, explore AAV target tropism, contrast in vivo versus ex vivo strategies, and wrap up with a decision framework.
Section 1. The delivery problem.
Finding the right delivery vehicle is completely indispensable for any successful experiment or clinical therapy. But before we can load our cargo into that vehicle, we have to decide what form the cargo actually takes. CRISPR components are typically delivered in 3 main molecular forms. First, as DNA on a circular plasmid. This enters the cell and requires the cell's own machinery to transcribe and translate it into Cas9. Second, we have mRNA paired with a separate guide RNA. Now, this skips transcription but still requires the cell to translate the mRNA into a protein. And third, the RNP, or ribonucleoprotein complex. This is where pre-assembled Cas9 protein is physically complexed with the guide RNA before it ever even touches the cell. The active enzyme is delivered directly. No transcription or translation needed inside the target cell at all.
Section 2. The 6 major platforms. And this brilliantly illustrates our biotech toolbox. Our options basically fall into 3 broad categories: non-viral, viral, and physical methods. We're looking at plasmids, RNPs, lentiviruses, AAVs, LNPs, and electroporation. And each of these vehicles has a completely different profile when it comes to kinetics, expression duration, and overall safety.
Section 3. The deep dive. Look, in genetic engineering, there's no perfect method, only trade-offs. First, let's contrast plasmid DNA with the RNP complex. Plasmids are cheap, there's literally no size limit on the insert, and they're super easy to produce. But because the DNA persists in the cell for days or even weeks, Cas9 just keeps cutting long after your desired edit is made, which dramatically increases the risk of off-target mutations. Plus, there's a risk of the plasmid randomly integrating into the genome. Conversely, the RNP complex is the gold standard for precision. Because you deliver the active protein directly, it has the fastest onset but also the shortest duration, usually just 24 to 48 hours. It does its job, and it degrades. This rapid hit-and-run style means it has the lowest off-target rate and 0 integration risk.
Next up, lentiviral vectors versus lipid nanoparticles, or LNPs. Lentiviruses are RNA viruses that integrate their cargo permanently into the host cell's genome. This is excellent if you want stable, long-term expression for something like a CRISPR screen in dividing cells. But for therapeutic gene editing, that permanent integration carries a severe risk of insertional mutagenesis, meaning it might accidentally disrupt a healthy, critical gene, like a tumor suppressor. LNPs, on the other hand, are non-viral. They're nanoscale lipid particles that safely encapsulate mRNA and guide RNA. They don't integrate into the genome, providing safe, transient expression. In fact, LNPs are highly clinically validated. They served as the absolute workhorse delivery platform for the mRNA COVID-19 vaccines.
Now let's look at AAV vectors and electroporation. Adeno-associated viruses, or AAVs, are small, non-pathogenic DNA viruses. Crucially, unlike lentiviruses, AAVs are non-integrating. They just sit as an episome inside the nucleus. They're incredibly powerful for targeting specific internal organs inside the body. Electroporation takes a totally different physical approach. It uses high-voltage electrical pulses to transiently blow tiny pores open in the cell membrane, allowing your CRISPR cargo to just rush in. It's a brute force method for sure, but it's highly efficient for hard-to-transfect primary cells sitting in a dish.
But before we crown AAV the ultimate winner for internal delivery, we absolutely have to talk about its Achilles heel: 4.5 kilobases. That is the strict maximum cargo capacity of an AAV particle. Cas9, the standard Cas9 protein we use, is massive. The gene encoding it barely fits inside this limit, leaving almost no room for the guide RNA or the necessary promoter sequences. This 4.5 kilo base constraint is a massive headache for researchers, often forcing them to use smaller Cas9 variants or really complicated dual AAV systems.
Section 4. AAV target tropism.
Now, what's really interesting about this slide is that despite that size constraint, AAVs remain incredibly popular. And that's because of their superpower, target tropism. They're basically targeted delivery drones. Natural variants of AAV, called serotypes, preferentially target completely different tissues. This tropism is determined by the specific viral capsid proteins on their surface. So if you want to deliver gene therapy to the central nervous system or muscle, you package your CRISPR cargo in an AAV9 capsid. If you're targeting the liver, you use AAV8. For the retina of the eye, you pull AAV2 from the toolkit. You choose the serotype based entirely on the organ you want to edit.
Section 5. In vivo versus ex vivo. So the crucial point is that understanding these vehicles brings us to the two ultimate battlegrounds of gene therapy. In vivo editing means formulating your CRISPR components and injecting them directly into the patient, allowing the delivery vehicle to find and edit the target organ naturally in situ. Ex vivo, also called autologous editing, works completely differently. You actually harvest cells from the patient, edit them in a lab outside the body, perform quality control, and then reinfuse those newly edited cells right back into the patient.
In the real world, for in vivo editing, we rely heavily on LNPs to target the liver, or AAVs to target the eye or CNS, but the ex vivo pathway is equally monumental. This is the exact strategy used by Casgevy, the very first approved CRISPR therapy in the world, which treats sickle cell disease. They harvest blood stem cells from the patient, and because these primary cells resist chemical transfection, they use electroporation to blast our gold standard RNP complex right into the cells. They edit the cells to reactivate fetal hemoglobin and then reinfuse them. The patient essentially becomes their own cell donor. Notice that for this historic CRISPR therapy, there is no viral vector used at all for the editing step.
Section 6. The decision framework. So what does this all mean? How does a scientist actually choose their weapon from this toolbox? Well, the therapeutic goal entirely dictates the vehicle. If you're editing primary human T cells to manufacture CAR-T cancer therapy, T cells resist standard transfection, so you must go ex vivo, using RNP electroporation. What if you want to edit a gene in a patient's liver to cure a disease like TTR amyloidosis? You go in vivo and you use an LNP, because LNPs naturally traffic straight to the liver via the ApoE receptor. Need to edit photoreceptors in the eye? You go in vivo with an AAV2 or AAV5 local injection for excellent long-term retinal transduction. The goal sets the vehicle.
And that brings us to the core motif of this explainer. Mastering CRISPR doesn't just mean understanding the Cas9 enzyme itself. It means mastering the delicate balance of these biological trade-offs. The delivery method you choose literally dictates the safety, the precision, and the ultimate success of the edit.
As we wrap up, I want to leave you with this thought. We've seen that the cell membrane is a fortress, and today our battering rams are precise but constrained. But with lipid nanoparticles expanding their targeting abilities beyond the liver, and researchers actively engineering entirely new viral capsids, the toolbox is expanding so rapidly. Could we soon see a universal, perfect, zero-trade-off delivery vehicle? The horizon of genetic medicine is moving fast, and the delivery problem is right at the bleeding edge. Keep questioning, keep learning, and thank you for joining me for this explainer.